Thanks a lot. Thanks for the invitation to the organizer. Thanks for organizing this great meeting. It's a really pleasure to hear so much about microbiome research. So yeah, this is a project that was done by my PhD student Roshi, who's now in industry, who is a very gifted person. And so I will talk about a method that we have developed that for de novo discovery of conserved microbial gene clusters. It's just in minor revision as Nature Methods, so we hope that we can get it out soon. Yeah, but there's a bioRxiv paper available. So as you probably all are aware of, we have a severe lack of functional annotation in microbial genomes. The best investigated microbiome is probably the human gut, and there we have 40% of the protein coding genes without any function annotated. Many of those that have functions annotated actually have mostly functions annotated from homology inference. So if this is referring to the core genome like KEG, then you also have information about the pathway. But if it's not in the core genome, so accessory genome, then mostly you might know that it's an N-nuclease or a transcription factor or what, but you don't really know what the protein is doing, in what process it's involved. So our idea was to help with this situation by finding functional associations using conservation of gene neighborhood. So the current tools, so why is gene neighborhood useful? Obviously because functionally related genes are often co-localized as conserved gene clusters, and we know this is true for operons, which are usually containing functionally associated genes. Biosynthetic gene clusters, microbial resistant genes, virulence islands, phage defense systems, and so on. And the reason is obviously that if functional associated genes need to be kept together to function, then this is a selective pressure that keeps them together. Yeah, so most tools that have tried to, or that have used this information detect only a specific type of cluster. For example, there are a lot of good tools for biosynthetic gene clusters, but few of these tools actually can do de novo discovery, and these tools are usually really slow. They use BLAST or other slow and also not very sensitive techniques, and most of them are very strict in the type of conservation. So basically they look for perfect or almost perfect synteny. So we wanted to address these So what Spacedust does is you give it a set of genomes. So what that means is that you give it the protein sequences, the positions, and the strand. Actually, this can be extracted by Spacedust from the genomes, but this is what it internally uses. This is the information it uses. And then it does a homology search with fold-seq using PROST5, or by looking up the structures through MMSeqs in a UniProt database if it can find them. So it does an all-against-all homology search using a very sensitive tool that was developed in my group, fold-seq, that uses structure and therefore has a lot of gain in sensitivity over purely sequence-based tools. So you have all these matches now between proteins in this set of genomes, and this could be hundreds or thousands. And then the key step is this cluster detection. So then we look for groups of genes or neighborhoods that have matching homologous genes. And then obviously these hits are reported. So let's look at this, how this cluster detection works. So here you have two genomes. Do I have a mouse? No. You have two genomes, genome 1 and genome, or genome Q, genome T. And a key to this algorithm is that we can...
If you click here...
okay, yeah.
You can use the...
Yes, that's good. Thanks a lot. So key to this algorithm is that we can measure quantitatively the significance of a certain local similarity or certain matches. And so we developed two p-values. One measures how strong the clustering is regardless of ordering. So for example, if you have these three genes within this square of 3 times 4, right? This is a relatively strong clustering, and it's very unlikely to occur by chance. So we calculate the p-value as the probability of this degree of clustering to occur by chance, and then we have an ordering p-value that looks at whether the order in these two genomes is conserved. So for example, here the order is perfectly conserved, although we have a gap here, and the strandness is also conserved. So the ordering p-value subsumes the order plus the strand. And from these p-values, we can simply calculate a match score. And now what we do is to find these clusters. So we have not only one such pair of genomes, but we have actually n squared pairs. So we look for every match. We try to extend it to a cluster that becomes significant by very just greedy search, trying to add another one and calculating the significance score, so the cluster match scores. And if it increases, then we take it. We take the best one available in the neighborhood, and by this we basically increase the cluster score until we reach the maximum. And so, for example, this one here would not be chosen because it has a strandedness or ordering p-value that is a bit lower than for the green cluster. Okay, so this is all there is. And so now we tested the tool by taking the 7,000 genomes from keck, reducing redundancy with 70% amino acid identity cutoff, running it with Spacedust, through Spacedust, and so we got 107 million pairwise cluster matches. And now we can look at a little bit of statistics. So 58% of all these 4 million genes in these 1,300 genomes had at least one cluster match, and here you can see how many matches you got. So for 58% of the genes here in green, you got at least two matches, and then you get fewer and fewer when you make the cutoff larger. So there's quite a big coverage. Those that are covered actually are very useful for predicting functional association, as this you can see here. So as a proxy for functional association, we use the annotation with the same keck pathway. Of course, only a small fraction of genes or it can be annotated with keck pathway for two genes that are near to each other. So I plus 1 or I plus 2. So it's a very good statistics, and you see that the precision, so meaning that the two genes are annotated with the same functional pathway, is very high for genes that have been part of cluster matches. So then we can visualize this. So here we have the genome on the x-axis, and you see that so the position on the x-axis and all the genes on the y-axis. This is the genome that we look at here in more particular, and you see these clusters here in 1, 2, 3. They actually did correspond to conserved clusters. Some of them are known, but some of them are unknown. And an example of an unknown one is, or there was unknown at least until 2021, is a CRISPR subtype E cluster, or type of clusters where we actually have this subtype 3 in our data. So we would have been able to find it in 2021, and we actually find not only the 17 subtypes discovered by the original publication, but also 7 others. Actually, 7 others in the data set. So we also benchmarked this. I have to now skip through this a bit faster. So we looked at how well Spacedust can identify antiviral defense systems, and there's a tool that can annotate them fairly well using a huge library of HMMs. And actually, so for most of these 106 defense systems, we can find most or all of the genes without any prior knowledge. So we have no HMMs, nothing. It's just de novo, completely de novo. So this shows you that most of these things can actually be discovered. And yeah, last slide is showing a comparison with biosynthetic gene clusters. So with Cluster Finder, DPGC, and Gecko, and you see that the F1 score of Spacedust is usually quite a lot higher than for these other tools. And here's a cumulative plot that summarizes this. And you see Spacedust here in blue has usually higher F1 scores. Yeah, and to summarize, so Spacedust is a tool for de novo discovery of conserved gene clusters. It's highly sensitive due to the structure-based fold-seek search. It's also very fast. can do all against all searches of thousands of genomes. It can find imperfectly conserved clusters. And lastly, it's open source, and there's a Google Colab notebook available. And yeah, I'm really grateful to Roshi for the great work, and also Milot helped a lot with the technical parts. Thanks.
Thank you very much. We have time for one quick question. Yes.
Thank you for the great talk. I wonder, when you're looking for the clusters, do you take a taxonomy of the genomes in which you're looking consideration? Like, do you weight?
No, we don't. So we basically assume that this is one, I would say, one limitation. We assume that genomes are far enough to be randomized, but this is something we want to work on. That if you put in genomes that are, let's say, very similar and not completely randomized, that the algorithm will correct for that.
Okay, thank you.
Thanks for the question.
So let's thank Johannes again. Thank you very much for this great talk.